Good morning. Today we will discuss about different staining techniques: simple staining, negative staining, gram staining, and staining of root nodules in leguminous plants. First, we will discuss about the simple staining. Simple staining required is curds. Inoculum is curds. We have to take a green slide, drop inoculum with the help of inoculation loop, and then smear the same on the slide, and then after that we have to make the smear air dry. Next is heat fixing. Heat fixing is done in order to fix the inoculum on the slide, which should not be washed in running water, so that the inoculum should be fixed on the slide. Then we have to stain with different types of stains. The stains used are crystal violet, methylene blue, or saffronin. Crystal violet we have to add little and then wash in running water. While washing in running water, we have to keep the slide slant. Then after that, air dry. Then we have to, the slide is ready for observation. When we observe the slide in the microscopic field, we can see different types of bacteria, rod-shaped bacteria and spherical bacteria. Rod-shaped bacteria, lactobacillus, and spherical bacteria, that is cocciform. This is about the simple staining. Next is negative staining. Negative staining is also known as indirect staining. We are using nigrosine as the stain here. We have to take the inoculum. Before inoculum, we have to take the stain here on the slide and then inoculum on the same. Then make the smear, air dry, observe. There is no need of heat fixing or washing. We have to air dry and then we have to observe. Indirect staining we are using, it is also known as negative staining. In this, mainly the microscopic field, the background will be stained and the bacteria will glitter. You can see the cocciform and lactobacillus, as we are taking the inoculum as curds. This is about negative staining. The third one is differential staining or the gram staining. We are using three different types of chemicals here. One is the stain crystal violet, the second one is modern gram iodine, and the third one is the counter stain saffronin. In between we are using alcohol for decolorizing. So here inoculum is taken on the slide, then smear is done. After smearing, heat fixing. Before that we have to do air dry after smearing. Then heat fixing is done. Heat fixing is done due to washing. The inoculum should not be washed off. Here crystal violet is added as a first primary stain, so that all the bacteria will get a blue color. Then washing is done after crystal violet. Then after washing we have to use the second chemical, that is grams iodine. So this grams iodine is called modern. It will fix the first primary stain. After that we have to wash it or decolorize. Agent is used, that is alcohol. We have to decolorize the stain. Then after decolorizing we have to use the counter stain, that is saffronin. After using saffronin, you have to keep the slide for a few minutes, that is 1 or 2 minutes. Then blot the slide, air dry it. The slide is ready for observation here. We are using crystal violet, we are using modern, then we are using decolorizing agent alcohol, we are using the saffronin as the counter stain. In this differential staining, when you observe observe after air drying, we can see the two types of bacteria here. One is gram-positive bacteria, another one is gram-negative bacteria. Gram-positive bacteria will take blue color, that is the first primary stain, crystal violet. The gram-negative bacteria will take the second stain, that is counter stain saffrony, so that gram-positive bacteria will have blue color and gram-negative bacteria will have pink color. The next one is about the root nodule staining. We have to follow the differential staining here. So root nodules are present in leguminous plants. They have the bacterioids in their root nodules. Here we have to take the root nodules, crush and take the inoculum on the slide. Then smearing is done. Remove the debris. Air dry. Heat fixing is done. Heat fixing. After that, we have to use the crystal violet stain, wash with, then follow the second mordant, that is mordant is used, iodine, then alcohol for decolorization, then counter staining, saffroning, blot the slide and air dry. The slide is ready for observation. So when you observe under microscope, you can see different types of bacterioids. Bacterioids you can see rhizobium. So it may be rod shaped or eye shaped, L shaped, etc. Star shaped bacterioids you can see. So this is about the root nodule staining, that is rhizobium is found, which is found in leguminous plants which are symbiotic bacteria.